Hi there. Today we'll look at BERTology meets biology, interpreting attention in protein language models by Jesse Vig, Ali Madani, Lav R. Varshini, Kaiming Xiong, Richard Socker, and Nazneen Fatima Rajani. This paper is an investigative paper into models that are trained on biological data, specifically into BERT models, actually into one specific BERT model that is trained on protein sequences. Now, it is trained to simply perform language modeling on these protein sequences. But out of this language model, you can then inspect this BERT model and read important biological data of these proteins, higher order data from the attention heads of the BERT model, which is pretty interesting. Basically means that the information of these higher order functions is at some point encoded in the structure of the language of the protein sequence. So we're going to go through what this means and how this comes about and what they did in order to investigate. I think this is a pretty cool investigative work and probably very promising for future research. Yeah, as always, if you like content like this, consider sharing it out and leaving a like. Also, tell me what you think in the comments. So, biology. Really quick for people who maybe have never heard this, in your every cell you have this thing called DNA, which basically is an encoding of all of your biological functions. Now, usually biological functions are realized through proteins. So DNA is basically a building plan for all of your proteins. This happens in the following two steps. First, there is this transcription step where RNA is built. This is basically a copy of your DNA, but is only single strand, as you can see right here. And then there is a translation step that finally translates the RNA into the protein. What will end up is just a sequence of these beads right here. Now, these beads are what are called amino acids. So a protein is simply a chain of these amino acids. There are 20 different amino acids, and the order of these amino acids in the chain makes the function of the protein. Now, specifically, we know about these proteins that it seems to be very important how their three-dimensional shape is. So a lot of these different amino acids have different chemical properties. Some are sort of, I think, negatively charged, some are neutral, some are acids, and so on. So they have very different chemical properties. So once you build this protein and you kind of release it into the cell, it will curl up into a three-dimensional structure. So this one might be, doing something like this and sort of form a circle or something like this, just because these proteins here, they kind of attract each other, maybe electrically, and thus the protein forms a circle, and the function of the protein is very much related to its shape. So if it is a circle, it could maybe, trap something else in here. So you really have to think of these things like kind of tools. There are proteins that cut other proteins, and they are really shaped sort of like a scissor that exactly fits these other proteins, such that you can effectively cut them. So sometimes you can substitute an amino acid for a different amino acid, like this here. If it doesn't change the shape, very often you're fine. The protein function isn't changed. But if you change a different amino acid that is sort of vital to the shape, and the shape changes, then your protein very often loses function. So mutations in DNA sometimes lead to mutations in protein, not always, because there is some redundancy in this translation step from RNA. But if they do lead to a different amino acid, it doesn't actually mean that the function changes. So there is sort of value in analyzing the sequence of the structure of proteins rather than the structure of DNA. Of course, it's also important to analyze the structure of DNA, but it is equally important to analyze the structure of proteins, because not all the information is in the sequence. Not all the obvious information is in the sequence. So what does this paper do? This paper goes and takes a model that has been trained on protein data. So if you look at this protein, it is simply a sequence of amino acids. And these amino acids, they all have names. I think I have a table somewhere here. Yes, so these are the different amino acids that exist. And you can see a protein is simply a sequence of these names. So usually they're abbreviated by like a 3-letter abbreviation or just a 1-letter abbreviation. So a protein might be A, V, M, M, V, A, G, and so on. And this is just a string of text, right? So what I can do is I can train a language model on this. A language model is simply a model that takes a piece of text and tells you what's the next piece of text. So what's the next letter, what's the next word? In this case, what's the next amino acid? And we can use tools from NLP for that. Specifically, we can train a BERT model. Now, BERT works a bit differently than a standard language model. BERT does what is called masked language modeling. So you would take this string, you would feed it into a BERT model right here. And I've made an entire video on BERT if you want to check that out. And what you'll do by inputting that, you'll mask out some of the tokens. So you'll maybe mask out this one, and then you ask the model to reconstruct those. We say that here's an M and here's an A without seeing them. So the model somehow has to learn from the surrounding amino acids what this amino acid could be, right? So it has to reconstruct this sequence. So the hope here is, in natural language, is that BERT somehow learns something about language itself. By being able to reconstruct these things, it has learned something about language, about which words appear together and when. It might even learn very long distance relationships between words, just because it has to predict those. And the idea carries over to biology. So we might hope that a BERT trained on an amino acid sequence will learn something about the language of proteins, about the amino acid sequence. And our goal here is to ask, can we somehow infer the 3D shape of a protein, which is the important part, from its sequence right here? So given its sequence, can we infer the 3D shape? Now, as I understand it, usually this has to be done in like a simulation. So you would build this in a simulator, and then you do like some sort of a molecule simulation to see how this ends up in a 3D shape. You could train a model to just predict the 3D shape, but in this case, we're just interested, what does the BERT model learn about the 3D shape while only ever having been trained on predicting the next or predicting the sequence of amino acids. So it's never been trained to look at the 3D shape. And that's our goal here. So specifically, we'll look at two different things. So here you can see examples of proteins and their high-level structure. So in these proteins, what you call the primary structure is this sequence of amino acid. This is simply which amino acid are in which order. There is a thing called the secondary structures, and we often observe that spans of these amino acids, like substrings, form these what are called these helixes, as you can see here, or these sheets. I don't know how they're strands in English. We call them sheets, or I think these are the alpha helixes and these are the beta sheets. And there's also turns. I think this here might be a turn. So there are these kind of secondary structures. And then the tertiary structure is how this is one protein. This is one unbroken chain of amino acid. And you can see this here kind of forms this double ring which would be its tertiary structure. Very important for predicting the tertiary structure is to predict when two amino acids are close to each other. So if we have a chain right here, and the chain, as we saw before, kind of turns and bends on itself, then these two amino acids here are very close, in close contact. And to predict which amino acids are in close contact to each other helps you determine the tertiary structure. So that's a consequence of it. So we wonder, does BERT know intrinsically which of these amino acids are going to end up being in contact with each other without ever having been trained to do it? The second thing we're interested in are binding sites. So here, well, you might not be able to see, but we made this example before where this sort of forms a loop, and then I say can trap something here, right? Like another molecule. And this is what we would call a binding site. A binding site is one amino acid that maybe through the structure of the surrounding amino acid as well, but also through its properties and how it is exposed in 3D shape, acts as sort of a receptor for other molecules. It binds to other things. It can, so think of your hemoglobin that traps the oxygen in your blood or something like this. It is where a chemical reaction or a reaction with something else will happen. That's a binding site. And we are interested, does BERT, the BERT that is only trained on a language modeling objective, know which ones are the binding sites? Because, you know, that would be very interesting and not something BERT was trained on. By the way, I particularly liked Richard Socker's tweet on this. I think he tweeted out, BERT trained only on language model can predict binding sites and biological properties, and formulated it like it was, you know, like GPT-3 was formulated. Like, if we train on Wikipedia, our model can do math. I thought it was kind of a satire headline. If we train on Wikipedia, our model can predict biology, and also it can tie your shoes and cook your dinner. Yeah, but so it's trained on language modeling on biological data, and now that makes sense. So they're going to look at two different things, or actually more than two different things, but they formulate this in an abstract way right here. So what they'll look at is the so-called properties, a property F. And this property F can be, for example, that a amino acid is a binding site. The property F can also be that two amino acids are in contact with each other. So F always takes I and J. If in the case, for example, where this is the contact property, then it simply is the indicator function for when I and J are in contact. And if it is just a binding site, then I think we are looking at J. At the token level property, we define to be an indicator that returns 1 if the property is present in token J. Okay, so whenever J is a binding site, then that holds. So what we're looking at are these attention heads in BERT. If you don't know, BERT has an attention mechanism, which basically means from layer to layer, each token can attend to all other tokens. So here, the amino acid sequence, I've drawn it twice, and the next layer representation of this amino acid will be able to gather information from all of the other amino acid through an attention mechanism, through a dynamic routing algorithm. I've made a video on attention is all you need if you want to find out more how this works. Now, what we're interested in is the strength of these connections. So the hypothesis is if molecule, here is 1, 2, 3, 4, 5, and 6. If molecule 1 and 3 are contact sites, then maybe we will find a layer where this connection between 1 and 3 is very strong, right? That would indicate that there is a connection site. Or that would indicate that BERT has learned something about the connection sites. Okay, if we find this repeatedly, so if we look at many proteins, and whenever we know that there is a contact between two things, and then we observe that the corresponding attention is very high, then we can be pretty sure that BERT has learned something about contact between amino acids. Okay. The same goes for binding sites. So if 4 here is a binding site, and then all the connections, all the attention that the higher layer gets from 4, so all the information routed away from 4 is very strong, that means all these other tokens are paying special attention to the token number 4, to this amino acid. And if we find that there's a big correlation with this being a binding site, then we can reasonably conclude that BERT has learned something about binding sites. All right, so we're going to do a correlative analysis for proteins where we know the binding sites, where we know the contacts, right? We can analyze them, we can run simulations, therefore we can know them. So we're going to look at this quantity right here, which is simply a normalized quantity. So we're going to look at the attention in a given attention head. So as you know, BERT has many layers with many attention heads. And we're going to look at whether or not this property is active and just normalize it by the total attention in that head so that we get some kind of a percentage number. That's the first task. We're basically going to look at how does the attention correlate with these properties. And the second task we're going to do is this probing task. So a probing task is like a linear probe in like a classifier. So what we're going to do is we're going to take a layer right here, and even though it's an intermediate layer, we're simply going to run it through a linear classifier and then decide, is this a binding site, for example, or not? Is a given amino acid a binding site or not? Is a given pair a contact or not? So this is kind of a linear probe, but this sort of takes a backseat in this paper. The analysis is really on the attention heads and what the attention heads learn. And that's already it. They take a pre-trained BERT model, so there are these BERT models that are already trained on these protein databases. And first, they look simply, can we find attention heads that correlate with a given amino acid? So here you see the attention to the amino acid. This is proline, I believe, and this is phenylalanine. Is that the same in English? Yes, phenylalanine and proline right here. So you can see that the plots here there's almost no attention pretty much throughout the network that pays special attention to the amino acid proline, except this head right here seems to have, if you look at the scale, over like a 70% of attention always goes to proline in this particular head. So this is layer 1, head number 11, focuses 78% of its attention on proline. Now this is not that special if you think about it, because in language models as well, in natural language models, you might want to think that you have some mechanism in your neural network that's especially specialized on like a very particular word in the language, because that might just be an often occurring very particular word. For example, in English, maybe the is very important, or the word what. These are like very indicative, very often occurring words, so it is reasonable to expect to find an attention head that pays a lot of attention to these things, especially here where our vocabulary size is 20 instead of like 30,000 in natural language. And the same goes for this phenylalanine, where you can see that in the last layer, and in the first layer you have attention, and also in the proline you have in the last layer. So why does this make sense? Because what we would expect from like single tokens, these are not interactions yet, these are not biological functions yet. So we know that in the lower layers of a neural network, we have these kind of basic features, basic feature extractors. And here, these basic feature extractors appear to be simply paying attention to one specific token in the vocabulary a lot. Okay, so they these heads sort of specialize for single amino acids. And the same in the last layer. So in the very last layer, the task of the very last layer is to prepare for the classification task. So if you remember the BERT model, you have layer, 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 and at the end you'll have to predict which ones are masked down here. So at the end you'll have to predict single amino acids again. So if there's a proline masked here, you'll have to predict the proline. So it also makes sense that the last layers would very much specialize to single tokens. So this does make sense. Now our question is going to be, do we find the biological function? Where would you expect them? We would expect let's say the secondary structures, which are sort of one level higher than the primary structures, we would expect to find them maybe here. And then we would expect to find the tertiary structures maybe somewhere here, okay? Because these are most highest level. And then it goes back again. Or maybe it's like we find the tertiary structures rather here and here again, and then in the middle we'll find the most high level, the tertiary structure. Sorry, yeah, blue secondary. This drawing is getting too weird. But there could be multiple scenarios, but that could fit here. But until now, it sort of makes sense. So they do an additional investigation where, as I told you, sometimes you can substitute an amino acid and nothing really happens, right? And in fact, this probably happens in you right now. You probably might have some mutation that changed some amino acid and you don't even realize because it's fine. No notice. So the biologists can build these matrices of how much you can substitute proteins with each other. So here you see this Blossom 62 substitution scores, which are very high if you can substitute two amino acids with each other, and the effect is negligible. And it's very low if it's the other way around. Now this is interesting so far, but you compare this to this matrix right here. This is the attention similarity. So what we'll do is, for each two amino acids, we take those two attention things, those two attention matrices, and we'll calculate the correlation between the attention matrices. And our hypothesis is that the more correlated the attention patterns are between the two amino acids, the more likely we are to substitute them. Because as a direct result of our language model is reconstructing, right, these things. So our language model is going to treat, if in natural language is like a synonym, right, our language model is going to treat synonyms very similar to each other because they're synonyms, they can be exchanged. So a good language model should learn that they are almost the same, and therefore the attention pattern is going to be almost the same. So a high correlation, we hypothesize, means that the function of the amino acid is similar, and therefore we can substitute it easily. So this here is the matrix of the correlations between each two attention patterns of these amino acid. And if you compare the two right here, they look extremely similar. Just have a look for a little while, and you'll see that the patterns, they do not match perfectly, but they are very, very similar. The dark spots are in the same places, the light spots are in the same places. So this already makes a good case that the language model here has learned something about biology. Now, what we want to do is we want to investigate higher order functions. So here, we're interested in these contact maps, right? So how likely is it that two amino acids are in contact? And we'll look at it through the lens of attention as we did before. So here you'll see percentage of each head's attention that is aligned with contact maps, averaged over a data set, suggesting that head 12.4 is uniquely specialized for contact prediction. So look at this. This head here is just spiking. So remember before we said our analysis is whenever we're basically measuring the correlation of two things being in contact, because we know it from our simulator or from our data set, the correlation of that with an attention connection being particularly strong. And we find it in this attention head right here. So this layer 12, head number 4, will always peak out whenever two things are in contact. Now you can see that it's not like always, it's like 25% of its attention, but significantly more than anything else right here. In fact, if you group the things by this attention, you can build the following plot. So you can see right here, probability two amino acids are in contact as a function of attention between the amino acids in head 12.4. Showing attention approximates a perfectly calibrated estimator, which would be the green line. So here we simply, for each pairs, two amino acids, for each pair of amino acids, we make a histogram right here of what their, sorry, not a histogram. We plot the probability if they have the attention weight 0.9, we plot how likely is it that they are in contact. So this is this. If we just look at the data and we simply take this attention weight as a measure, as a predictor of being in contact, we get the blue curve. And the green curve would be if we could perfectly predict from this attention head what the probability of contact would be. And you can see that the fit is fairly good. You can't predict with super high accuracy, but the fit is fairly good. And you can see the general trend that as the attention in this head rises, the probability of the two amino acids being in contact with each other also rises. So we can sort of confidently say that BERT has learned something about a higher level biological structure just from the language modeling objective. How can we interpret this? This must somehow mean that it is possible, it is vital for reconstructing the sequence from its surroundings. So if we delete this right here, if these two are in contact in the 3D structure, that makes probably means that this thing right here is a very good predictor of what was here, right? If we mask this out and we're asked to reconstruct which amino acid was there, then it probably helps to look at its neighbors, right? It probably always helps to look at one's neighbors, especially also in natural language. But if these two are in contact, they have very special connection to each other. You can basically read out from this one which one this was. This is sort of like if you have a sentence and you say, does, I don't know, I can't come up with one right now, but if it's like da 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 da, and then there is a name like Mark, and then da 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 da, and then there is him, right? And you would expect if I drop out, okay, let's do it the other way around. If I drop out him, then from the text right here, you can probably determine that it is some sort of pronoun. But then you go back and you see, it's Mark. Okay, so it's not like it or some or she. It's probably he or him. This is sort of the analogous structure right here in biology. The second thing we're looking at is these binding sites. Now, these are single properties of different amino acids, and we're simply looking at all the incoming, or sorry, all the other tokens that focuses their attention. Why is this important? Because these binding sites are central to the structure of to the function of the protein, right? If this here is a binding site, then that's a very central, important point of the protein. So a lot of these other things are going to be determined by what this binding site is. This binding site needs to have a very particular function, and therefore probably needs to be a very particular amino acid. And the other things here are sort of supporting this binding site because they form the 3D structure around it and so on. So you would expect a lot of attention to be put on this binding site. And what do we find? We find that it's a bit more murky than before. So you can see that the attention is kind of spread out. Percentage of each head's attention that focuses on binding sites. Especially in the deeper layers, binding sites are targeted at much higher frequency than would occur by chance. Head 7.1 has the highest percentage with 34%. So also here you can see that it is spread out, but this is because multiple heads are now focusing on these binding sites, because probably binding sites come in different variations. So you'll have lots of heads specializing on attending to binding sites. And they say it is much higher frequency than would occur by chance. And you can see here, this head is the highest with 34% of its attention focused on binding sites. You can also see the general trend of the attention being rather in the later layers, which we would expect from a tertiary structure. Now, yeah, it would be interesting here. Here you also see that actually most of the things are in the last layer, which points to rather maybe lower level information, because we reasoned before about the last layer, or I was just wrong. But also in a general trend, you can see that the attention is rather shifted towards the later layers, because this is sort of a higher order function. Okay, if you look at the same calibration experiment, you can see that the picture is not as clear. There is the general trend at the beginning, but then it sort of flattens out. So you can sort of differentiate the very probably not a binding site from the somewhat probably a binding site, but it's not a perfectly calibrated classifier. And that might just be because there are many things specializing in different types of binding sites. So you can't just go to this one head. So this is just for this one head. You can't just go to that one and expect that to classify all the binding sites, because you might want to combine all of the high-ranking ones here to form a classifier. The last experiment they do is these linear probes, where they just go and they just build classifiers from different parts of the network. And you can see right here that what is predicted and how well they work. So each bar here is going to be the difference of performance. So this is differential performance of diagnostic classifier by layer, sorted by task order in figure 8. Each plot shows the change in performance between the given layer and the previous layer. Okay, so a bar up shows it's performing better than the previous layer, a bar down shows it's performing worse than the previous layer. So you see right here that these are the secondary structures right here. And you can see that there is a lot of performance in the earlier layers right here. and sort of not that high performance in the later layers. Whereas for the tertiary structures, the binding site and the contact, you can see that there is a bit of performance in places, but it sort of tends to be more towards the middle, certainly more towards the middle and the end of the network than the secondary structures, which sort of makes sense with our hypothesis. You can also see this here, where they show the percent of attention focused as a function of layer, and the red is the center of mass, and you can see that as the secondary structures, their center of mass is at a lower layer in general than the tertiary functions. All of this is not perfect, of course, but it's still an open question, I guess, whether or not it's not perfect because we haven't built a strong enough language model yet. Do I want to say GPT-4 is now for biology and not for language? Or is it because there's really, you really can't very well predict these things just from a language model? I mean, you should, technically all the information is there, but maybe the language model objective as such isn't able to capture that information. So yeah, this was the paper. It's pretty simple. They have in the appendix, they have a lot of these additional experiments or full experiments, I believe, for all the amino acids and so on. And I invite you to check that out. In general, I like this kind of work because it's very applied. It's and it can, tell us something about the nature of both these language models and the biological things that we care about in biology. Okay, I'm just talking crap right now. Thanks for being here. I hope you enjoyed it, and bye-bye.